started this morning with a recorded version of What You Get Is What You See by Tina Turner, which is the theme song of Assumption Zero. Now, I'm sure that none of you slept last night because you were just thinking about PACT. It's the most important thing in your entire life right now. So, this morning I'm going to I'm going to redo the second part of yesterday. And there are a few additional things because I want you to really have a feeling for the sense of the approach. And then at the end today, I'm going to add some new things to show you how this connects with larger theoretical issues about evolution as a complex systems phenomenon, evolution and complexity theory, and things like that. So, from yesterday, this new algorithm, PACT, integrates these three critical elements, which are all necessary if we're going to provide accurate historical descriptions in evolutionary biology. When any kind of evolutionary phenomenon has produced simple patterns, that is, when there have been relatively simple, vertically transmitted kinds of evolutionary phenomena, PACT gives exactly the same results as any other methods, all the other methods that have ever been produced. And in PACT, we call this the template. Now, this is a sort of a conceptual thing to help you begin with the method. Normally, what the template is, if you're doing a PACT analysis, you have a group of trees, at least three, and you just arbitrarily choose any one of them as your starting point, and you call that the template. That's just the beginning point. And then as you add information to it, add more trees to it, you just build the tree as you go. Now, if the second tree that you add is identical to the first tree, then the template doesn't change. The diagram, the PACT diagram doesn't change because all the information that's in here is contained in here. So you can add this to this without having to change the original diagram. And then this is just an indication of what you could do to check with yourself. This is also what a computer program would do. You may also have diagrams like this. You may have historical records, pieces of history that are not exactly identical to this because they don't include everything that's in this original. But everything that is in this one is included in here. So there is an A at the base, and there's a group called CD. And so those are the two pieces of this diagram. They're already in that diagram. So that means you can add them together without changing anything. And the same thing is true if you have lineage duplication here, because this is identical to that, this is identical to that, this is something different, but it's down here somewhere. So those three categories, those three classes of evolutionary outcomes can all be combined in a way that doesn't change your basic template, doesn't change the basic answer. These are simple processes, and as long as your data set only includes these kinds of phenomena, it doesn't matter what kind of method you use, you will always get the same answer. And that's basically what we call in PACT, we call that combination rule 1. Anything that looks the same is the same. In other words, if you can combine things without changing your template, then you just combine them. However, and it's not just biogeographic patterns, but when any evolutionary patterns show complex outcomes or the result of complex phenomena, then you begin to get differences between what PACT does and what other methods do. And it's at that point when we start adding realistic amounts of complexity into the data, that's when we begin to see progressive differences in the different methods of analysis. So for example, in this particular case, as before, there's only one difference between this, and that is that this diagram has something new in it. There's something unique in this diagram that's not in that diagram. Now what PACT does not make any judgments about explanations. PACT is just describing. So that means that you never eliminate something. So if there's something new here, you add it to the existing analysis. And you never remove something. You just add things to it. You build a tree sequentially. So this would be your new template. You have to remember what all of your input trees are, because at the end of the analysis, when you begin to try to explain what's going on, you have to remember that this was something unusual, that this was something unique in one group. And then you have to explain that. But as far as making the tree, making the PACT diagram is concerned, it doesn't matter. And this is the second combination rule, which is if you have a template and the new information includes information that's in the template plus new information, you just add the new information. And this is an example of the same kind of thing, except in this case, it looks like it's a different kind of phenomenon because the new piece of information here that's being added is associated with a taxon or an area or a host or a population, whatever your token is, that's already in the diagram. And this is what we call a reticulated pattern. And in this particular case, the computer has two choices. Do you associate this A with that A, or do you associate this A with that A? Well, it's a really easy thing. If you put this A here, the entire diagram aligns, and you just add that new piece of information. And so your new template looks like this. And that's now a combination of 5 different trees. And this is also, even though it looks like it ought to be something different because it's now a reticulation, as far as PACT is concerned, it's just new information. So PACT doesn't make any value judgments. PACT doesn't assign any kind of explanation to different kinds of observations. It's up to you to assign explanations after you've done the analysis. So this is pure description, and it's the same combination rule. If you have new information in the tree you're adding to your template, you just add that new information into the template. Now, we went through this before, and we'll see if it makes any more sense to you today than it did before, because this is one of the observations that causes the most trouble for people who are just learning this. And you notice that there's a lot of there's a lot of mismatches between these two diagrams. And in fact, the only thing these two diagrams have in common is that they're both anchored by this thing called A. Other than that, they have nothing in common. Okay? So the PACT result is this. And the fact that there's a trichotomy here, a polytone here, means that there is no information that tells you anything special about the relationship between these and these, except that both of them are connected to that in some way. So it's a possibility that that's reality. And as I showed yesterday, the one possible explanation for that would be something where in biogeography, this would be a common source for biogeographic dispersal. And this would be one set of areas that represented one episode of dispersal and speciation. And this would be another set of areas. So you would have a situation perhaps where you had a mainland and 2 sets of islands in 2 different directions. And you have one set of dispersal here and one set of dispersal there. And what you end up doing is you produce this evolutionary event with that as the sister group or the ancestor, and this set of evolutionary events that also has the same source. So it's possible that this represents reality, that there's something explanatory about this, in which case we would call this a hard polytomy. On the other hand, it's possible that this just means that we don't have enough information to say anything, in which case it's a soft polytomy. It just means get more data. Now, get more data is always a good thing. But in this particular case, it's a slightly new combination rule. It's a modification of the previous one. So we have this little progression. Y plus y equals y. Y plus yn equals yn. Yn plus yn equals ynn. Okay, so these three combination rules actually are hierarchical, but they have one thing in common. You're always adding new information to the tree that you're building. You never take anything away. And that's one of the reasons it doesn't matter where you start, and it doesn't matter what sequence you use. For one given set of trees, you will always end up with the same final result. Now, the intermediate stages may look different because of the sequence, but the final result will always be the same. And as we saw yesterday, there's still the possibility of some ambiguity when it has to do with unique events. But that doesn't actually change the robustness of the tree, and it doesn't change any of the interpretations of what the general parts of the pattern are. Now here's an additional situation where you have these what's called paralogous distributions or paralogous or setups. And this is a case in which you can attach that A here or this A here. And in either case, wherever you attach, whether you attach this A with this or this A with this, you're going to end up with the same diagram as a result. So you don't have to make a decision, because this is fundamentally a decision about age. Is this the same age as that, or is this the same age as that? PACT makes no judgments about that. PACT just describes the data. So, again, you just add that information together. And this is a combination, the fourth combination rule, which has to do specifically with paralogous distributions of information, where you have two things that are the same but are paraphyletic. And it's important to have this combination rule because it turns out in all other methods, including both primary and secondary BPA, those paralogous things would be combined and collapsed. And it turns out that is an assumption. It's one of two equally parsimonious assumptions, explanations about parology. But it's not, because it's not the only assumption, because there are two equally parsimonious interpretations. It's not permissible to just choose one arbitrarily in your method of description. So it turns out that what we have are 6 different modules of pattern. So all of these area cladograms, host cladograms, all these historical diagrams, cladograms, branching diagrams I'm going to show you, all of them are combinations of only 6 different pieces of pattern. We have the first module where there's 100% congruence. The second module where there's something in the new tree that is not in the new tree, that is in the original, in the template. Something where you have lineage duplication. Something where there is new information in the new tree being added that does not produce a reticulation. Something in which there is new information being added that does produce a reticulation. And then finally, there's this issue of parology. So, of course, because all you did was obsess about PACT all night and none of you have slept or anything, you have this one burning question, which is, if there's 6 modules of combination, why are there only 4 combination rules? If there's 6 modules, why are there only 4 combination rules? That's because 3 of those modules are the same thing. The difference between something missing from the new tree and something extra in the new tree is just a matter of the sequence in which you add them. And whether the new information, produces a reticulation or not, it's just added this new information in the same way. So it turns out that all three of these pattern modules, as far as PACT is concerned, they're all the same thing. So that means that PACT really only recognizes 4 modules of patterns, 4 classes of patterns, all cladograms, all phylogenetic trees, all gene trees, all host cladograms, all area cladograms, for everything, everywhere, all the time, are combinations of only 4 modules of pattern. Okay. So now I'm going to do this again, which we saw yesterday, and this is an attempt to try to visualize what you do intuitively. You do this intuitively, and then this is how you check it. These are the comparisons you make, and this is what the computer would use as data, but you're able to actually do these, to superimpose diagrams, because our brain is better than any computer with respect to comparing complex patterns. Both of these are exactly identical, so this is the combination, the first combination. Here, the patterns are identical, except that we have something missing. Okay, there's nothing. I did that right? No, this is something added. This is something extra. Sorry. And that's if there's something new added in. And then this is the one where there's something missing. There's something not here. But that's still an example of adding in. So you have y plus y equals y. Y plus yn equals yn. Yn plus y equals yn. Okay? And it doesn't, that's what you can see, that in fact, these are just different faces of exactly the same thing. And here you have something, both something missing and something new. So the template has EF, the new thing has EG, so the new thing has something extra, and it's also missing something that's in the template. So, because this one has something new, and it's unique so far as you know it right now, this analysis, you don't throw it out because it appears to be unique. You add it in because it's new information. And because this does not appear here, you don't throw it out because it suddenly doesn't appear on new data. You never eliminate observations. You only add new data. And this is a yn plus yn equals yn, so you end up with this trichotomy. And then finally, we have the parology rule here. It kicks in. This is the fourth combination rule. That, and you end up with this as your template. And now you add another new tree to it, and this one has something new connected there that creates a reticulation, but it's still an example of y plus yn equals yn. And that's how we see that all three of those modules of something new, something missing, something new and not reticulated, something new and reticulated, all use the same combination. There are just different manifestations of the y plus yn equals yn rule. And here we have one case in which you can actually collapse something, where you have a terminal that has something duplicated. You can collapse that, but you don't have to collapse it. Okay? And it's possible that in some cases you may have additional complexity in here that makes you not want to collapse that. But if this was the end of your analysis and that's all the data you had, you could collapse these into 1D, but you would still have to remember that in explaining your data, this 1D showing up there for one of your clades actually represented a duplication within that entity. But as far as PACT is concerned, it doesn't matter whether you collapse that or not, as long as it's in a terminal branch and you don't have any other data. But in general, if something's homologous or something's duplicated, you don't collapse anything, you don't combine anything unless it's way down there and sort of outside. So now here's an 8. So the tree that we've got now, this template back here, that's a combination of information for 7 different clades, and only 2 of them are identical. And they were only identical at the very minimal level. So this is a combination of information. This diagram, there is no one clade that actually shows that entire history. And in this particular case, what we have is something after an H attached to B. In this diagram, there's only one B, so now we have an interesting case. We have the one category of cases in which you can end up with an ambiguous solution in PACT, and that is this B can be connected here or here. Now, the default PACT, and I'll reiterate that later, the default PACT is to always, when there's ambiguity, always make the connection with the most recent possible solution. And that's based on the assumption that if we have no other information, any ambiguity will be resolved in favor of the most recent period in time. And that's based on the assumption that unless we have evidence to the contrary, we expect to have better information about events in the recent past than events in the deep past. But no matter where you put this, you can either put this here or put this here, you will end up with 2 different trees. But then, whoops. Sorry. I somehow dropped a slide. The final result here is you have two different trees. Okay? And they're only different in terms of whether H is here or H is there. So in circumstances like that's the one kind of situation in which PACT can end up with multiple equally parsimonious outcomes. But the thing that's interesting about it is that this really only pertains to unique pieces of PACT. If you had a number of other results that supported this, you would end up resolving it in one particular way. But this is just to let you know that it is possible for evolution to be so complex that no method of describing the data will be able to resolve all of the ambiguity. This just resolves most of the ambiguity. Okay. So why is PACT better than anything else? One of the reasons is because of the way it deals with the problem of horology. All other methods, including both primary and secondary BPA, would take these two and combine them into one. And that amounts to saying that we believe the only way this could happen is through sympatric speciation. Okay? Now, the so-called pattern clades, the people who believe that you can do phylogenetic analysis without evolutionary mechanisms, don't call this sympatric speciation. They call it lineage duplication, as if lineage duplication does not imply some kind of mechanism. But the mechanism of lineage duplication, if you're talking about gene trees, is gene duplication. Lineage duplication, if you're talking about species, is sympatric speciation. Now, it's also possible that you can get this without lineage duplication. You can get this with dispersal. Okay, so this could be the result of horizontal gene transfer. It could be the result of hybridization. It could be the result of geographic dispersal. And it turns out that one episode of geographic dispersal versus one episode of sympatric speciation are equally parsimonious. So your method of description should never make an a priori decision about which is correct. That's for you to decide when you go to evaluate the results, your discoveries. But your method of discovery cannot make a decision for you or for the data. So you have to understand that anytime you have this in your PACT diagram, you have two different possibilities to explain. And it is not the function of the method of description to do your work for you. It's not the function of PACT to explain your data. It's your job to explain your data. The job of PACT is simply to describe your findings. It is a method of pure discovery. Now, in addition to that, there is one set of circumstances in which combining paralogous entities in trees and then combining the trees will actually obscure information. It will actually make it appear that there is no information when there really is some. In this particular case, here are two trees. If you combine them with PACT, this is what you get because you maintain that paralogy. All the other methods, including secondary BPA, combine those two, and basically it just says, well, it's AB, we have AB, so the result is actually this. Now, this appears to be more parsimonious than that, but it's not more parsimonious, it's just simpler. There are three entities here, there are four there. 3 is more parsimonious than 4. 3 is simpler than 4. But it turns out that this is actually more parsimonious in the sense that if you test both of these diagrams with new data, the simpler one collapses. And the more parsimonious one is the more robust one. So if you take these two diagrams and you add that new tree, PACT continues to give you the same answer. And so this result basically says that all three of these trees represent 3/4 of this. And each one represents a different 3/4 of that overall diagram. But if you take this and this and this and combine them using all other methods, they tell you that there's no information there at all. So this is the most parsimonious description of the data. This is the simplest description of the data. This one has no information in it, and this one has information in it. So this is a case, as I said before, this is a case in which simplicity is not parsimonious. And I'm going to return to that at the end, and it's really, I want to really get you to understand this difference between simplicity and parsimony. And this is a problem with every single method that has ever been produced, including mine. And it's because the way we analyze these things with a matrix of information, we end up combining everything. We make the assumption that everything in there was associated at the beginning of time. Nothing came in secondarily. So we start by pulling everything together at the base, and that forces us to explain all ambiguity as arising later in time. So this is like some sort of Aristotelian utopia or Platonic utopia, where once upon a time the world was perfect, and through time things changed, and every time things changed they became more complex, and things got worse and worse and worse. And the equivalent of that is, originally, everything was simple. And then it got more complicated. And what we want to do is eliminate that complexity and keep the old simplicity. And it doesn't matter whether you're talking about historical biogeography or socioeconomic development. It's the same human mentality. And it turns out that this is a hidden assumption of mechanism. And making an a priori assumption about mechanism biases the outcome. No good method of description of discovery can have any a priori prohibitions on possibilities. There can't be this kind of bias inherent in any discovery process. That's why any method of analysis that begins with a model is not a method of discovery. Okay? When somebody does Bayesian likelihood, they're not asking, what do the data support in terms of a phylogenetic hypothesis? What they're asking is, given that I'm assuming that this model of evolution is true, what is the most likely true tree for the data? Now, if we interpret the data in this way, what tree does it support? That's not the same thing as saying, what tree do the data support, and what model does that then imply? So there's two very different ways of looking at the world. Now, it turns out that if you end up with the same answer, approaching things in these very different ways, this is really good. If you can get to the same answer using very different approaches, then that's very nice. We feel then that the answer is very robust. But oftentimes we don't get the same answer. And then instead of saying, we need to look at this in a different way, what we do is to say, my model is better than your model, my gene is better than your gene, morphology is better than molecules are better than... Or in the case of some cases I know in the Journal of Parasitology review process, well, one of these studies was produced by Americans, the other was produced by Mexicans. Well, we know the Mexicans must have made a mistake. Even though it turned out, in that one particular case, it turned out that the only reason the two results were different was because the American group had misidentified two species. And when you correct the species name identifications, both of the results were identical. But the Journal of Parasitology never published a correction. Just a small social comment. Not that you've ever run into any kind of angle bias in your lives. Okay, now there's another thing that PACT does. PACT also eliminates the problem of parology. The other quote-unquote problem that Nelson and Plattman identified in what they call holistic biogeography, which was the basis for all these a priori methods, the ones that use assumptions 1 and 2 or particular models. It's what they call the widespread taxon problem. Okay, that is, what happens in biogeography when you have one species that occurs in more than one area? Oh, this is a big problem. Okay, assumption 1 takes care of that problem. You just eliminate the information about the distribution in areas that do not conform to a vicariance hypothesis. And then you say, it was all secondary dispersal. It's okay to throw that information out. The problem with that is that widespread taxa are actually evidence. They are not noise in the system. They are not like an error term in an analysis of variance model. And the reason they're evidence is because without dispersal, you cannot have vicariance. Vicariance is based on the assumption that you have a widespread ancestor, which is then subdivided by climatological or geographic alterations. So this has always been a logically inconsistent part of the entire holistic biogeography program. And they have recognized that that's a potential internal inconsistency, and their explanation is, well, that's permissible because we know that dispersal does not produce general patterns, and because we're scientists, we're only interested in general patterns and not unique events. Science doesn't explain singularities, like the Big Bang, and therefore, it's okay for us to throw out information about unique events. And we know that dispersal doesn't produce general patterns. And the reality is we all believed that until very, very recently. And in fact, it's one of the errors that Deborah McClellan and I, actually one of the very few errors, that we perpetuate in the nature of diversity relative to phylogeny, ecology, and behavior, which was published in 1991. Even in 2002, in the nature of diversity, we repeated that mistake. That general patterns are the result of vicariance or isolation, whereas dispersal doesn't produce general patterns, so we don't have to worry about it. We can document it, but we don't have to worry about it. It turns out that there are two mistakes in that. The first is that dispersal, widespread attacks are evidence. They're not noise. They're not epiphenomenal. They're actually part of the explanation. And our own examples in the nature of diversity show that dispersal is really important. Even though we were repeating this thing that dispersal doesn't produce general patterns, the examples that we show, like when we reanalyzed Joel Craycraft's Australian bird data and showed that it was not one pattern of vicariance, it was actually two patterns of dispersal coming into Australia, and then one lineage, one group of clades dispersing around the west coast, another dispersing around the east coast, and everybody meeting down in southern Australia for a party. That's what it actually showed. But we were still repeating the mantra that, oh, only vicariance leads you general patterns. And that's because we did the proofreading of the nature of diversity in early 2001. And then turned the book in, and it was being printed when I discovered Bruce Lieberman's paper from 2000 saying, hold on, cowboy, there can be general patterns of dispersal, and here's how they can happen. And by that time, it was too late to change the nature of diversity. But since then, we've been talking about the fact that you can actually get general patterns out of dispersal. And that changes everything. It changes how we view these diagrams. It means that you can't just say, here's a PACT cladogram that has general parts and unique parts. You ignore the unique parts, and all the general parts are vicariance or isolation or whatever your token is. You have to be more sophisticated about how you actually begin to interpret the diagram. And that's because you actually can have general patterns of diversification based on horizontal processes. You can have, if you're a botanist who works with a composite, you understand that there's an enormous amount of hybrid speciation that's going on. If you're somebody who works with the biogeography of oceanic islands, you understand that there's been a lot of coordinated colonization. There are general patterns of island colonization. And here's an example. These are two trees, and the letters just stand for, let's say, islands in the Caribbean. If you combine those two data, those two trees, with every method that doesn't allow duplication, it says these trees have nothing in common. There is no information there. PACT says, not so fast, cowboy, there's actually a lot of information in here. It's just information about colonization. And the reason you get structure here and you don't get structure before is because here you take both of the trees and you combine them here and then look for generalities, and there isn't any. And what PACT does is PACT says, oh look, this group started from A, this group started from F. These are two groups. In fact, this turns out to be a group that colonized the Caribbean from Florida, and this is a group that colonized the Caribbean from Venezuela. So they came into the Caribbean in different ways, but once they got to the Caribbean and got on the same islands, after that, they participated in some shared dispersal and colonization of other islands. And I pointed out yesterday that secondary BPA takes these two from the diagram and moves them down. This one's up here, and this is up here in the PACT diagram. And it turns out that the reason for the different placements between PACT and secondary BPA is that what PACT says is when there's ambiguity about where you place a taxon, you always maximize combinations starting with the most recent. So what PACT does is it makes all the ambiguity down in the oldest observations. And that's, like I said, this is the same thing as saying if there's ambiguity, it's more likely that we have better information about more recent things than about older things. And all the other methods end up forcing us, just because of the way they begin the analysis, the way they route the cladograms, they force us to adopt the alternative view. That the older the observation, the better our information about it. And that's probably not true, although it could be true. It could be true. This sounds more reasonable to me, but it's not necessarily true. Okay, so it turns out that this is the PACT default. There are other approaches. And if any of you know anything about character optimization on phylogenetic trees, this is equivalent to what's called the Deltran optimization. And this is equivalent to the Actran optimization. The Actran optimization is the one that's favored by most phylogeneticists. And Wiley, in his 1986 paper about assumption zero, suggested that Deltran would be better for historical phylogeography because it favors dispersal over extinctions. But it turns out that it's actually a slightly different reason to prefer that. It's because we believe that the more recent the data, the better preserved the information is. That's really the reason. So one of the ways to look at the difference between secondary BPA and PACT is to think of PACT as something like secondary BPA that uses Deltran rather than Actran. That's, if you know anything about these other methods, earlier methods, that's one way to make the transition. But in fact, it's much better to just discard all of that previous 40 years of development, except to recognize the importance of people trying out different ideas, eventually to try to get to some stable solution. But that's where we are now. We have a stable solution. Sociologically, it's not being adopted, but that's just sociology. That has nothing to do with the actual science of the situation. So what happens if we have that kind of ambiguity about placement? Well, the interesting thing is that it doesn't change the PACT diagram. It changes your interpretation of where individual clades and individual parts of clades might be placed on the diagram. So that means that it's not part of the method of describing the data. It's part of the method of explaining the data. And that means that you have to go and look at individual clades, and you have to look at individual clades in the context of co-occurring clades. Whether those clades, those trees are gene trees or phylogenies of species, you have to do what Billy Henning called reciprocal elimination. You have to have a minimum of 3 trees in PACT in order to begin to explain. Now you can do a PACT description with one clade. Okay, if you want to do PACT phylogeography with one clade, you just remove the names of the species and put the geographical areas on the diagram, and you're finished. That's the answer for PACT. If you combine 2 trees, you can get a PACT result, but you don't have, when there's ambiguity, you don't have any way to choose whether you have, for example, if one of the trees has something the other tree doesn't, and you only have 2 trees, you have no way to decide whether presence or absence is the special condition. You don't know whether there's been secondary loss or whether this is something unique added. So you have to have a minimum of 3. And in fact, what you really need is more. You need as many as it takes. With PACT, you need as many clades as it takes to get an asymptote in your sampling saturation curve. And that means you keep adding clades until you no longer get new pieces of pattern. And at that point, you have sampled enough of the clades in the area, enough of the parasites in the host, enough genes in the genome, enough populations in the species to say that you have the answer. That you expect that everything you find subsequent to that will be part of this pattern that you've discovered. You also need to interpret the nodes. If it turns out that we not only have general and unique nodes, which we've always thought about, but in the past we just eliminated everything, all the unique stuff, and said, we don't have to worry about the unique stuff. That's not part of the explanation. But what we have not recognized until Bruce Lieberman's paper and subsequent, his book and his subsequent articles, is that we have to interpret the general nodes. The general nodes are not necessarily just vicariance nodes. They're not even necessarily just speciation nodes, isolation nodes. That there actually can be expansion nodes, that is horizontally transmitted nodes, as well as vertically transmitted nodes. So that's speciation and dispersal in biogeography. That's co-speciation and host switching for host parasite systems. That's lateral gene transfer and horizontal gene transfer, vertical gene transfer in the case of things like gene trees. And Bruce came up with an approach to this which uses character optimization on phylogenetic trees as its background. And there are a number of different ways to do this, but they tend to be fairly robust in our experience with respect to most of these trees. So here's an analysis. You have this thin line, that's unique, that's unique, that's unique. So you still have to provide 3 explanations for the 3 unique events. But for analyzing the nodes, what you're asking about are the general features of these diagrams. Okay? So just because I say you don't count these, when you're analyzing the nodes, it doesn't mean that they disappear. It doesn't mean that you throw them away. The thing about unique events is they require unique explanations. And what the cladistic biogeographers and the cladistic people have been saying is unique things have no explanation. Therefore, they don't exist. Therefore, we can throw them away and ignore them completely. Therefore, we don't have to count each event of throwing something away as a cost of our analysis. Therefore, we can pretend that making up a lot of extinctions is not less parsimonious than just allowing some host switches or some dispersal. So that's not allowed. These are real data. In biology and evolution, unique events require unique explanations. General observations require general explanations. And in this particular case, so you have, here's this node, this is a CD node, the C and D are connected. That node connects down to a B. There's nothing shared, so that's, you just keep adding. You go down here, there's yet another one in it. So you end up down here with ABC, BCD, CD. So what you have is from one node to the next through time, each of these general nodes connects a smaller number of areas. That's what happens when you have isolation events, speciation events, or vertical subdivision of genomes, evolution of subdivision of genomes. This is that example I showed you before, where the H can be here or it can be here, there or there. Now it turns out that that only affects how you explain H. It doesn't explain, it doesn't affect the rest of the PACT diagram. It doesn't change the rest of the analysis. And in this particular case, because that's a unique event, when you're analyzing the nodes, it doesn't matter where it's placed. Because this, and this are all going to require special explanations. and the general node analysis on this diagram and that diagram are the same. So that's an indication, again, of the robustness of the PACT results. And this is a more realistic situation. This is one in which you go from 2 areas to 1 to 4 to 3 to 2. So this is isolation, expansion, isolation, isolation. So this is how you can find alternating episodes of expansion and isolation. Or here is the dispersal that allowed this vicariance. You don't have to be afraid of widespread taxa. The presence of widespread taxa will not obscure the vicariance patterns. It actually highlights them. It makes them more robust. So all of this fear of complexity in the development of these phylogenetic methods has been misplaced. In fact, you end up, if you try to eliminate dispersal, you end up working against yourself in some way. It's also true that when you reanalyze all the published data that have been published as examples of all vicariance, and tomorrow I'll go through a number of classic cases of this, it turns out that when you reanalyze them, this is what you find. You actually find that half of the general nodes are expansion nodes and half of them are vicariance nodes. And the signal is not a signal of vicariance alone. It's a signal of alternating expansion and isolation, which is not the maximum vicariance model, it's the taxon pulse model. Like I said, we'll come back to that again tomorrow. So, not all general patterns are due to isolation, and not all isolation patterns are due to vicariance. Peripheral isolate speciation can produce general patterns when you have a common dispersal corridor from a given source area to a particular island, for example. And you'll end up with the same pattern, even if the events don't happen synchronically, that is, at the same time. As long as they all occur within a time between changes in the environment when the colonization, the dispersal corridors are no longer available. So again, we come back to this thing, where you get the general area cladogram. We use Lieberman's protocol, and it turns out there are only 3 general nodes. But here's what happens. This is, one group comes in from Florida, one group comes in from Venezuela. It takes them some time to get together because they come into different islands. But once they meet in the middle, once they get together in the Caribbean here, then they disperse together, and then they get isolated together. And some of them continue to colonize. So this is a picture, again, this is the most parsimonious representation of the data, but it's not simple. And it allows you to find a situation in which we don't believe that both clades were associated with each other from the beginning. In fact, these 2 different clades don't get associated with each other until this point in history. Up until here, they're colonizing from different areas into different islands. And then they get together at this point. And from this point on, we have a coordinated history. But that history is still a history of dispersal and isolation together, plus independent dispersal events. And you can get all of that from a diagram that's based on using only 4 combination rules. So, when nature's simple, PACT finds simplicity just like everything else. When nature's complex, only methods of use of duplication rule are capable of finding the complexity, and of those, PACT is the best we have. Now, pattern modules. PACT recognizes that there are only 4 fundamental categories of pattern in historical trees. There are only 4 kinds of pattern. And yet, they can generate arbitrarily high degrees of complexity. The simplicity is in how the diversity is generated. It's not in the results. That's been the fundamental mistake that we've been making up until this point. We were looking for simplicity in results, and we were using progressively more and more complex methods of generating simple results. So now we have all these likelihood models and all this incredibly complicated mechanism for generating the results. But it's not simplicity of results that we want. What we want is an accurate description of the true complexity of the results. And what we want to find is the maximum amount of simplicity possible in the mechanisms that produce those results. So, for example, this has to do with pattern nodules on trees, but let me give you a parallel. There are only 4 nucleotides in DNA. That does not stop DNA from producing arbitrarily high degrees of complexity. The simplicity is in that generating algorithm. Those digital replicators, that expanding information space that I talked about in the first lecture I gave in Curso. It's not in the results. What makes the study of the origin of the genetic code interesting is that the basic building blocks are not very complicated, and the mechanism isn't very complicated, and the results are incredibly complex. That's what makes it fascinating. That's what makes evolution really interesting and different. So this is where we've gotten off track. We have been trying to force ourselves to have simple results. And because nature is really complex, the only way we can produce these simple results in the face of increasing evidence of complexity is to create more and more complex algorithms for forcing the data into simple results. So as Indiana Jones said, we've been digging in the wrong place. We've been doing things backwards. That's why PACT is important. It's not like I said yesterday, I have no idea that PACT is the best there will ever be. I do know it's better than anything else that's been produced up to this point in time, and I do know that if we believe that evolution is a process of generating complexity, that PACT is the appropriate discovery tool for all historical comparisons in biology. And so we'll take about a 20-minute break for coffee and bathroom and getting some air. And then we'll come back, and from here on out, the rest of the week, I'm going to be talking about different levels in the biological hierarchy, the genealogical and ecological hierarchies, different levels in which we can apply PACT to answer evolutionary questions. And I've already suggested in my first presentation in Curso that the only thing you need to produce arbitrarily complex hierarchical structure in biology is to have information that is encoded digitally and combinatorially. And that's exactly what DNA is. It's another example of how a relatively simple mechanism for generating outcomes can produce arbitrarily high degrees of complexity. And so our job is to accurately describe the complexity and then explain it. Our job is not to try to eliminate complexity from our lives because we're afraid of it. So even scientists have to understand that we are deeply, historically, conservatively afraid of complexity. Fear of complexity is in our brain stems. And we have to use the other part of our brain to overcome that fear and confront the reality. And it's actually not that frightening. It's not as frightening as we thought it would be. People who thought that if we allowed complex explanations in phylogenetically related areas, we would just lose control of everything. We would just be telling stories, there would be no general patterns. They were afraid. They were afraid of what complexity would do to their beautiful, pristine, simple worldview. It was just fear. And they don't have to be afraid anymore because we can handle this. Okay, see you in about 15 or 20 minutes. On this, I want to do one last thing on the board. This is what I should have had in the presentation about this combination. This is the way I should have explained it. So here are the two trees, and you number the nodes. Okay? Now there are three things you could do combining those trees. If you combine like this, you create a node here that's not in any of those trees. So this is overgeneralizing the data or over-explaining the data. On the other hand, if you just collapse everything, you've eliminated these two nodes. So that's undergeneralized. And what PACT does, because remember I was talking about being really focused on accurate representation of the complexity. This is too much, this is not enough, this is just right. Each one of the nodes is represented on that tree. Okay? So that's, if you were having trouble visualizing that combination rule, this is what I should have said. And the next time I give this lecture, I will have some slides that show this. Thank you very much for pushing me on this. As I was saying at the coffee break, one of the reasons I teach is because that's the best way for me to learn. It's the best way for me to continue to learn. So this is not an altruistic exercise on my part. I get something out of this also. I learn. Okay, so now we're going to talk about how do we use PACT to actually investigate interesting parts of the real world. Okay? And the first thing we're going to do is talk about PACT and genealogy. Now, in Hennick's work, Billy Hennick's work, he distinguished between what he called tocogeny and phylogeny. And tocogeny was tocogenetic relationships for Hennick, simply meant relationships among populations within species, as inheritance patterns among populations within a species. And there's some very famous diagrams that Hennick had of species lineages with all of this reticulated stuff going on inside it. And phylogeny was the genealogical relationships between species or among different species. So this is basically what we could call patterns of gene flow or patterns of information sharing among populations within a species, and this is patterns of relationships among species that are produced from common ancestors. And what Hennick said was, you can't do this using my method. You can only do this. And so in a sense he was saying, if you're analyzing some data and you can't make any sense out of it, maybe you're not dealing with different species. Maybe what you think are different species might actually be one species. And so this was kind of his way of giving people advice on how to decide what really is one species. But it's very general, sort of, it wasn't very explicit, let's say. And Hennick kind of thought this was self-evident. So he didn't really have any specific, he just sort of mentions this at the beginning and then goes into phylogenetic analysis. One of the people who has always been interested in whether or not there was phylogenetic signal in population level phenomena, that is, genealogical phenomena within species, is Ed Weil. And if you look at his 1981 textbook, he reiterates what Hennick's position, but then he says, but maybe that's not necessarily true, and what Ed was doing, in a sense, Ed was anticipating the entire discipline of phylogeography at that point. His You know, in the 1980s, when Ed would say, I wonder if there are any phylogenetic patterns in populations, there was this instant rejection. Population biologists didn't want to have anything phylogenetic in their work, and systematists didn't want anything to do with population biology in their work. And so basically everybody rejected this, even though the population geneticists now have their entire own set of phylogenetic analyses of population relationships. They're all very statistical and so on. Now, there were two papers published in 2004, one by Hafner and Riddle, one by Marshall and Sykes. And they said very much the same kind of thing. They started with this distinction between tocogeny and phylogeny. And what they said was that if you're going to try to distinguish different species from differentiated populations within one species, one of the things you should expect is that there should be a lot of articulations among populations within species and no articulations between species, populations that represent different species, or at least the level should be much lower. So we expect that information sharing among populations within a species is at a much higher level than information sharing between species. So even if you believe that there's been a lot of hybrid speciation, you don't necessarily think that those events happen all the time. They happen rarely. And so you expect that you should see lots of, if you see lots of articulations, you're within a species. The problem is that they didn't have a methodology based on phylogenetic reasoning for looking at this. So then they went on and said, okay, so we have all these statistical things like nested plate analysis and so on, and somehow we'll be able to figure this out. Isn't this interesting? But it turns out that PACT actually allows us to look at topogenetic relationships. And it turns out that there are a couple of reasons we might want to do that. So here's an example now. What we have here are 3 gene trees. So these are trees, basically phylogenetic trees of populations, and the letters represent populations. And these are gene trees based on haplotypes in different populations for 3 different genes, or the same populations for the same species or more than one species. And so here's what you get. These are basically haplotype maps. These are histories of haplotype differentiation or patterns of relatedness among haplotypes for 3 different genes within the same populations of some fish that we have always thought was one species. So what you would expect is that if you combine all those, you should see lots of articulations. If it's all one species, then there should be a lot of articulations. Well, it turns out, I mean, look how there are lots of duplications here. So there's a lot of evidence of articulation. But there's an interesting thing about the patterns of articulation. There are actually 3 independent lineages here. And the reticulations are all within the populations within those lineages. So we see a lot of evidence of reticulation, but we don't see any evidence of reticulations between these 3 lineages. So that tells us that in terms of information sharing among populations, there are actually 3 groups of populations here that are acting like 3 separate evolutionary species. So this gives us an additional criterion for trying to decide whether we're dealing with one species or more. So this is the way to actually test what this Hafner and what Hafner, Riddle, and Marshall and Sykes were saying about topogenetic relationships. In this particular case, it appears that what we're dealing with is actually 3 different species. Now, the interesting thing about this is that this particular species that's It's a little fish in North America. And the species name is inconstans. Okay, when you see species names that are things like variabilis or inconstans, that's almost always an indication that it's more than one species. Almost always it turns out to be a species complex. And it turns out that the real data for this is just an abbreviated version of this particular study. And unfortunately, the student who did the study never published it. But it turns out that if you look at the whole study that was in her dissertation, there is one place, there's only one case where you have to never happens in Harry Potter. There's one place where there's actually a reticulation between two populations in two lineages. And the only place that happens is in a place called Algonquin Park, about 150 kilometers, 200 kilometers north of Toronto. And the only place where members of two different lineages actually come together and share information is a place where, until about 40 years ago, it was near the origin of two different drainages that went this way. But about 40 years ago, that area was modified. And now those two drainages share some connection. So the only place that there's actually hybridization going on now is a place where there was relatively recent anthropogenic modification of the environment. Now we don't know what that's going to mean. It may mean that now that, we know that these things are acting like, or have been acting like, independent evolutionary species, to the extent that there is genetic differentiation and there's communication, strong communication within, or cohesion within the lineage. But we don't know that these had ever become species. We don't know that they had become irreversibly split from each other. And now that we do have this anthropogenic connection between two of the lineages, we don't know if that means that these will now collapse back into one species. That the initial conditions for speciation were established, but anthropogenic change in the environment actually collapsed that now. So we don't know what's going on, but that would be a place to go and watch and see what was happening. Because in this particular case, if you have two populations in two differentiated lineages that now have come together, either because of human modification of the environment or natural modification of the environment, then it's possible to watch what's happening. And one of the things you might expect to see is that information from this lineage would begin to make its way into this lineage, and information from this lineage would make its way into there. Because if you do have this kind of articulated sharing of information among populations within a species, then once you have information introduced horizontally, no matter where it's introduced, you expect it to end up being shared with all the populations in the species. Now, if it's not shared with all the populations in the species, then it's possible that the hybrids are actually at some fitness disadvantage. So there's a lot of potential information you can get out of, a lot of really interesting research projects you can get out of seeing this kind of pattern. Now, the opposite sort of situation would happen if you had, let's say, and let's just take these two for the moment. And let's say this is the pattern that you see. Okay? And what you're actually looking at are two different river systems. Say these are two different rivers that are tributaries of a larger river. And what you have is a species of fish, like salmonids in North America, that always return to the natal stream. They return to the river where they were born, to spawn. So They're out in the larger river or out in the ocean for most of their lives. When they reproduce, they come back in and then go up their navel stream. Now, one of the things you might expect in a situation like that is that the farther up the stream, up the river, the fish goes to breed, to reproduce, the less likely it is that it's going to have any interactions with a member of the other lineage. Down here, if they're breeding close to the mouth of the river, that's where you would expect to see the largest number of mistakes. If a species makes a mistake and goes up the wrong river to spawn, it's probably because the spawning site where it was born is close to the main river, is close to the mouth of the river. So if you have something like that, if you were investigating the possible divergence, the genetic consequences of very high fidelity return to the nesting site, what you would expect to see is that at the base of the tree, the base of the PACT diagram down here, that's where you would expect to see the possibility of directional mistakes. You would expect to see the largest number of articulations in the populations that were closest to the mouth of the river. And as you go farther and farther away, you would expect these diagrams to show more and more isolated differentiation as you go farther and farther up the different rivers. So that would be your prediction. Now, if it turns out, what happens if it turns out that you have nothing but isolation as you go up the rivers, and then way up here and way up here, you suddenly find populations that show some evidence of hybridization, some evidence of articulation. Then you have to start thinking about the possibility that way up there is some kind of stream capture. Or something else is happening that maybe you don't know about. But the a priori prediction would be that most of the reticulations would be in populations that nest close to the mouth of the river. Because you would expect that those would be the fish that would be most likely to make a mistake in what stream they go back to, because you would expect the conditions here and here, if that's near the mouth of the river, you would expect those conditions to be most like each other and most like the main river. So that would give the fish the least amount of information to distinguish between their actual original nesting site and similar nesting sites close by. Whereas if you go farther and farther upstream and away, you expect the fish to have more and more habitat-specific information to deal with. So you can just use your imagination. You just say, all right, here's the study I want to do. What would that look like? What kinds of gene trees would I expect to see? What kinds of relationships would I expect to see? Now, one of the things you have to remember about this is that, okay, this is just what I said, 33 populations, 2 deep splits. Now, the genetic divergence data suggested that there are, using this magic 5% rule that the barcoders use, that would suggest that there are 3 different genetic species. Now, that's just a typological species concept. It's the same as using morphology to tell you what your species are. But it actually turns out that because there's only one case where there's any reticulation between populations in these different lineages, it also looks like the PACT analysis makes it look like these different populations are acting like separate evolutionary species. And then this sets up this whole business of, have they completed speciation? Is it possible that these species, these incipient species, will all collapse back on each other if there are anthropogenic changes in the habitat? What's the basis for the differentiation? This sort of situation where you find sort of the opposite, where you find way up at the headwaters of this, way up at the headwaters of this. That's where you find the genetic exchange, and we know that that's because of human alteration of the landscape. But in a more natural situation, if you're talking about the potential divergence of two populations into two species, each one restricted to a particular river, then if you're actually catching speciation in the act, you would expect to see down near the mouth of the river the largest number of mistakes, the largest number of breeding mistakes. So you would still maintain some sort of gene flow down here, but as you go farther and farther up the river, you would expect to find more and more isolated divergence. Now, if you use nuclear genes in producing your gene trees and doing your PACT analysis, then the reticulations you find are not just evidence of geographic overlap, actually evidence of gene flow. That then allows the possibility of adding a historical component to standard population genetical accounts of gene flow. You actually have the possibility of saying what genes are flowing and in which direction. Which are the older, which are the younger. There are all kinds of things we could ask that we've never asked before, because nobody ever thought there was a possibility of recovering phylogenetic conservatism in population dynamics. But when you have exchange between populations, is it symmetrical or is it asymmetrical? Are the genes that are exchanged old genes or new genes? Now, there are lots of theories, lots of models, lots of expectations. All of these can be tested now directly with real evidence. And that's pretty cool. Well, one of the things that we want to know is whether or not the results of a PACT analysis for phylogeography actually coincide with the results of the nested clade analysis. Because if it turns out that a phylogenetically based assessment and a statistically based assessment give you the same answer, you have a party. So that would be really, really nice to know. And the other thing that we want to know, ultimately, is whether or not, once you've established what's going on within a species with respect to gene flow in populations, then you want to ask whether or not the same kind of population dynamics are happening in the sister species that lives in a different place, and the next species out, and the next species out. So, for example, are the population responses to selection and the proportions of gene flow and the kinds of gene flow phylogenetically conservative within clades? So does a given clade of birds, for example, have a clade-specific population dynamics profile? So that if you know the population dynamics of one species in the clade, you know the population dynamics of the other species. And if one of the species in your clade is very common and another species is endangered, you can make inferences about the population dynamics of the endangered species, and that may help you understand why it's endangered based on the population dynamics of the species that's common, its closest relative. So this is the way you can use these kinds of phylogenetic analyses and the assumption of phylogenetic conservatism to make policy, to make conservation policy, biodiversity policy. Now, parasitologists already do this. We just don't acknowledge it. We don't even understand why we do it. But we do know that if we're dealing with malaria in Brazil, we can use information about controlling malaria in Africa or Asia to help control malaria in Brazil. That's exactly the same thing. It's just supposed to be something magical because it's medical and public health. It's just good applied evolutionary biology. We just don't call it that. But that's what good epidemiology is. It's just good predictive population biology. The epidemiologists don't like to hear that because they're special. I had this happen to me one time. I made the mistake of saying in a lecture that was full of Harvard School of Public Health people that, yeah, after all, epidemiology is just good population biology. Oh my God, it was just, that almost shocked me, you know. Oh, that was completely different. Oh. Well, it's not completely different. This is just all sociology, over-specialized professionalism. You can't call yourself an epidemiologist because you don't have a piece of paper that says you're an epidemiologist. But the reality is that a lot of what we do in things like public health are actually unconsciously systematics and evolutionary biology. And they work to the extent that important epidemiological components are phylogenetically conservative. Transmission dynamics, life cycles in parasites or any pathogens, extremely conservative phylogenetically. And when something's conservative phylogenetically, we can make predictions. In biology, the only predictions we can make are predictions about the conservative nature of the past. And it's what philosophers call retrodictions. We don't make predictions about the future in biology unless we say the future is going to look very much like today. Okay? You don't predict what, if your sister's pregnant, you don't make predictions about what the baby's going to look like. You make retrodictions. You say, I'll bet the baby's going to look like you or your husband or their parents. And then the baby's born and you go, oh look, the baby just looks just like the father or just like the grandfather. Okay? And yet, that baby's going to be a unique person with unique traits. All the unique traits are unpredicted. Everything that was predicted is a recurring historical trait. It's a persistent piece of history. So you didn't actually predict anything, you retrodicted. But that's the power of phylogenetics in applied areas like conservation biology and public health. That's how we make use of it. We just don't acknowledge often enough that that's what we're really doing. So we really need to know this kind of stuff. You can't do proper biodiversity work. You can't do proper conservation biology. You cannot do proper epidemiology without evolution. And as the pharmaceutical companies know, you cannot justify producing drug-resistant strains of pathogens without evolution. So you need to have natural selection help you produce those little moneymakers. Okay. Now, we're going to go from population biology to actual phylogenetics. And now, for the first time, I'm going to show you a situation in which PACT is going to create new problems. Or, as graduate advisors call them, new opportunities for research. So here are things that happen between species. These are things that Henning said don't happen. But these are things that do happen between species. And they produce reticulated relationships. And if you don't want to have this in your phylogenetic analysis, you have to figure out some way to eliminate that kind of information from your analysis. Now, it turns out that one of the best ways to eliminate all of this from your analysis is to use a method of phylogenetic analysis that simply will not allow reticulations to appear. I remember an early meeting of the Billy Hennig Society when Garrett Nelson got up, and that was his position. Just reticulations are bad for cladistics, therefore we're just not going to recognize them, and they're not important anyway. And there's a room full of botanists listening to this dribble. And Ed Wiley got up and said, well, I've always thought that the best way to alienate people is to tell them that they cannot do something they've been doing for 50 years. So the reality is that botanists know about all this stuff. They can see it. The plants are incredible. If you have hybridization between two genomes, you don't have some combination genome. Oh, genomes are there. You look inside the cells and they're going, hi. I mean, botanists know about this stuff. And Ed's position was, if you don't accommodate that, the botanists are not going to buy into phylogenetics and they're going to go do something else. And here's the problem. Here's a phylogenetic tree that a botanist would recognize. You have 3 species, A, B, and C, and a 4th species, Z, that is a hybrid. It's a hybrid of A and B. Now, we're living in a perfect world. And in that perfect world, the hybrid species has the apomorphic traits of both parents. This doesn't always happen, okay? But it happens enough. So you have a situation in which if you just made a table of the characteristics of these 4 species, you would have species C would have the derived trait for character 1. A, B, and Z would have the derived trait for 2. A and Z would have the derived trait for 3. And B and Z would have the derived trait for 4. You get a little data matrix. You do a standard phylogenetic analysis, and this is what you get. 2 different trees, and 4 different optimizations of characters, 4 different interpretations of character evolution. That's what you get. 2 of the trees say that Z is the sister species of A. 2 say that Z is the sister species of B. In one of them, you have the convergent evolution of the derived trait for 4. In one of them, you have a secondary loss. And over here, you have the convergent evolution of the derived trait for 3, and here you have a reversal secondary loss. And that's what you get. Now, imagine that you have a reasonable sized database for a group of composite plants. Let's say 20 species, 10 of them produced by hybrid speciation, 10 different combinations of parental. What kind of phylogenetic analysis do you get? You get stacks of equally parsimonious trees. You get thousands and thousands and thousands and millions of different trees. Now, you can reduce that to 1 tree by using a model. And this is one of the reasons that there's so much interest in things like Bayesian likelihood and maximum likelihood models, because those models allow you to manipulate parameters according to your experience and insight after you have manually aligned the sequences according to your experience and insights to get the answer you feel really comfortable with, which is, I get 1 tree instead of 12 million trees. Okay, and that 1 tree may be the true tree. Now, I'm not saying it's absolutely going to be wrong. What I'm saying is it's not tested. It's not an independent description of the data, and it's not a testable product of anything. It's just a statement. If this model of evolution is correct, then for these data, that's the most likely tree. Thank you very much. So, how do you test that? Well, you don't. How do you know if it's right or not? Well, I've been working with this group for 30 years. I know what the relationships are. That's what let me adjust the alignments of the sequences. Because one of the little secrets is that there are no computer programs to do complete automatic alignments of sequences. Every single sequence alignment program we have is an approximation, and then you are expected to fine-tune the alignments by hand. And yeah, this has always been a little bit too nebulous for me. This has always worried me. And again, it's not because I think that the results are wrong. It's because I think the results are slippery. The results are not, you can't grab them and test them rigorously enough. But in the case of real data, where you have real hybridization, this is exactly the mess you get. Now, if you're doing DNA sequence analysis, you'll already have a mess because of the nature of the data. Okay, with DNA sequence analysis, since you only have those 4 little starting points, those 4 little modules of morphology, in any one site, in any one gene, by the 3rd mutation rate, the 3rd mutation event, the probability that the next mutation will produce homoplacy is 100%. So here's the trade-off. With macromorphology, what we call morphology, you don't have very many characters, but because each one is the product of integrated gene, multi-gene function, they tend to be very stable in terms of phylogenetic analysis. Whereas with sequences, you get millions and millions of data points, but each one of them is very unstable because they're so simple. And the probability of convergent evolution is so high in each one of them. So I have this trade-off. Okay. What would PACT do? What would Mike do? Let's go back now. Let's take these 4 characters and these 4 species. Okay? Now here's what happens. Here's character 1. The apomorphic trait is found in all 4 of the species. Character 2, the apomorphic trait is found in those 3 species. Character 3, character 4. Just do a PACT analysis. These plus this give that, add this, that gives that, add that, that gets this. So there's your PACT result. And what have you got? Well, you've got one species that shows up twice in a phylogeny. That can't happen. Species only evolve once, right? Species only have one ancestor. Yeah, unless they're hybrids, then they have 2 ancestors. And we only recognize monophyletic groups in phylogeny. Yeah, unless we have hybrid species, in which case, whichever group we classify the hybrid species with makes the other group paraphyletic. So, if you want to be non-arbitrary and actually evolutionary about your classifications, then every time you have a species that's a hybrid, you classify it with both parents. That species shows up in a classification in 2 different places because it had 2 different origins. Now, if this is just a matter of shared genes and you don't actually think that there's a hybrid speciation event. So, for example, let's suppose that Z really is the sister of A, but has acquired some genes from B through horizontal transfer, through accidental hybridization. But that hasn't actually produced a speciation event. If you know that, then of course you're justified in taking that out of your data set. But you have to test for that. And you're not going to test for that unless you have something like this that says, you have something to explain. So if you just have this, the assumption has always been you need more characters, or you need different characters, or you need a way to weight the characters. So let's find a model that gives us the right answer. Okay? So that's because people try to include explanation in their discovery methods. But in the terms of PACT, PACT just says that is the description of the data. Now, this could be the result of hybrid speciation, or it could just be the result of lateral gene transfer. Let's go find out. Let's test for that. We can test for that, and we know exactly where to go, here and here. So this is testable. PACT isn't going to explain it. PACT is going to describe the situation, and then you have to go and collect more information to actually explain it. Now, this is for what we would call unordered states. Okay, this is sort of standard morphological analysis. And the reason you know what the apomorphic trait is because you've done your nice outward comparisons and everything. What if you have gene trees? What if you have what are called ordered states? So here we have 2 gene trees. And we do a PACT analysis, we get the same answer. So if you have one gene that shows the connection between B and Z, and another that shows the connection between A and Z, that is, each of the genes represents 1/2 of the hybridization event, the hybrid speciation event, then PACT will give you the right answer, quote unquote. It will tell you that there was hybridization and there was equal genetic sharing. happens if there's hybridization, but you don't get this kind of equal sharing? What if this sharing is all asymmetrical? Well, then you don't end up with a reticulated diagram. So if you have hybridization that doesn't produce an actual speciation pattern, then you have an asymmetry in terms of information sharing. And that's one of the ways that you can tell that this was lateral gene transfer, horizontal transfer, and not an actual hybrid speciation event. Okay, now, here's the problem that I've just created for us. Standard phylogenetic analysis assumes that all homoplacy is the result of independent evolution within lineages, either convergent evolution or evolutionary loss, evolutionary reversals. That's standard phylogenetic analysis. You get the most parsimonious result. Everything that's inconsistent with that is homoplacy. And that's all expected to be the result of independent evolution within independent lineages. PACT interprets all homoplacy as the result of horizontal transfer. So here's what we've got. Standard phylogenetics does not allow horizontal transfer. PACT does not allow convergent evolution within lineages. Now, who thinks that the world is made up of only this or only that? Nobody. Therefore, Houston, we have a problem. Or, for all of you youngsters in the audience, here's a research opportunity. And I'm even going to tell you where to begin to solve the problem. We need additional information to decide when we use assumption zeros. If we invoke assumption zero, we're saying that it's hybridization. We're saying that it's horizontal transfer. If we don't invoke assumption zero, we're saying this. Okay? If there is any kind of hybridization, hybrid speciation, horizontal exchange of information, then assumption zero is more fundamental than parsimony. But it's not 100% true. It's just operation descriptively more fundamental. But it's not necessarily true, and it's undoubtedly wrong in some cases. So we need some method. We need additional information or some methodological approach to help us decide. Now, it turns out that we already have that. And Vicki Funk, who's a botanist at the Smithsonian, actually published the way to solve this problem in 1985. And nobody paid any attention. Now, what did she say? Well, what she did that nobody wanted to pay any attention to was Vicki wrote a paper saying, we need to look for patterns in the homoplace. We need to look for correlated homoplace. We need to understand that sometimes homoplacy gives us general patterns. Nobody wanted to hear that because we were still thinking, okay, the only general patterns of character change you can get are homologies. And if we allow homoplaces to be correlated, then phylogenetic systematics collapses. Now, Anne Wiley, this particular paper was published in a volume, a symposium volume, in the Annals of the Missouri Botanical Garden. I had a paper in there that used the term historical ecology for the first time. Ed Wiley had a paper in there. Joel Craycraft had a paper in there. Don Rosen had it. There's a classic issue. It was basically the beginnings of the expansion of phylogenetics from systematics into more integrated areas of biology. And so Ed and I listened to this. Most of the other people there were, no, get your crucifix, get some holy water. We don't hear anything about this. And Ed and I were looking at it. And I remember Ed turning to me and saying, this is something we've got to think about. This is something we have to take into account. And what Vicki said was, here's what I've done. said, for the last 2 years, I have been having people send me specimens of plants for groups where they know from cytogenetic studies, they know which species are hybrids, and they know what the parental species are. And I've had them send me specimens of every member of the group without telling me who the hybrids were. And then I do a cladistic analysis. And then I try to predict to them who the hybrids are. And she said, I'm correct about 80% of the time. Okay, and here's what she was noticing. Now at that time, we had to, all you youngsters, you won't even believe this, but once upon a time, if we wanted to do a cladistic analysis, we took a large stack of cards called punch cards, and they had to be, there are like 2,000 of them, and not one could be out of order. And we had to take them over to the computer center and put them in something called a card reader, and it would go, and then the computer would compile that program. And then we'd take another stack of cards, and those were the cards with the data matrix for each species. And we put them in. And half the time, you would just get an error message that said, did not compile. And that meant that 1 out of 12,000 cards in your program was out of sequence, and you had to figure that out. But if you were lucky enough, and go, program compiles, phylogenetic analysis, come back in a week, and you get a printout like this. And this is what was happening in the botanists. They were getting like 12,000 trees. And they were killing a lot of trees printing out all this stuff on paper. And what Vicki did was she started looking at all those output trees, and what she discovered was that there weren't, when it said there are 12,000 equally parsimonious trees, oftentimes there were only really 8 different tree topologies. And then there were all these differences in character interpretation on them. And she started looking more closely at that, and she found that this is what you often get. It's a situation, here's a situation where there's one class of equally parsimonious trees that have species G as a sister species of C, and another where you have this species G as a sister species of F. And there are 2 things that happen. The first is that if you remove G from the analysis, you get 1 tree. So that's your first indication with standard phylogenetic analysis, that there's something about that species that's screwing up the analysis. And the other is that when you look at the printouts, look at this. Oh, lots of homoplacy in that one species. Lots of homoplacy in that one species. And it turns out the patterns are different. Sometimes you get a whole bunch of correlated homoplacy on an interior branch, but that was Vicki's key to where the hybrids were. And so she would look at something like this and say, species G is a hybrid between C and F. And then she would send that off to the person who sent the materials, and they would say, how did you know? And when she was getting to about 80% correct, she decided that she would talk about this at a meeting. And she published the paper, and it was promptly forgotten. And it was an absolutely brilliant insight. But only now, 30 years later, almost 40 years later, are we at a point where we understand how brilliant that insight was and how important it is. This is where to begin. If you want to know when to use assumption 0 and when not to use assumption 0 in a phylogenetic analysis, you go back to Funk's 1985 paper and you bring that up to date. Now, it turns out that this does not always work exactly right, because when you have hybrid speciation, the hybrid species does not always show all of the apomorphies of both the parents. In a lot of cases, it doesn't. But this is the place to begin. And then there were some subsequent papers by Lucinda McCabe looking at the details of this, and she published some papers saying, well, you don't always get all the apomorphies of both parents, so you should be careful about this. But this is the place to begin. So what happens if we take these data for these species and do a PACT analysis? We end up with no homoplacy. See, because PACT doesn't allow homoplacy. All the homoplacy is explained away by the duplication rule. In this particular case, if G is a hybrid of A and F, then there isn't any real homoplacy. There's just hybrid speciation. On the other hand, if G is not a hybrid, then PACT analysis is giving you the wrong answer. And we have to ask, what's the reason for this? But the PACT analysis will say either G is a hybrid of A plus F, or G is the sister species of A or the sister species of F and has received significant genetic information from the other species by lateral transfer. So, of course, one of the first things you want to know is whether or not the two potential parents are sympatric. Or if they're not sympatric, are they wind-pollinated species and one lives at higher elevation than the other? Or something like that. I mean, there are all kinds of interesting possibilities for how you can get this. So the important thing is this doesn't give you the truth, but it gives you a description of the data showing some unresolved complexity and additional possibilities that traditional methods don't let you see. They don't allow you to see them. Okay. Now, PACT and genealogical relationships. Here are some questions that would be really, really nice to have people begin to investigate. When is it topogeny and when is it reticulated phylogeny? Now, when are these different populations within one species, and when is it hybridization between species? Okay. Now, if you go back and review your notes or the online, go back and look at the lectures from 2 weeks ago about speciation, this is critical to understanding whether you're dealing with parapatric speciation, whether you're dealing with speciation by reinforcement. If you want to study the evolution of isolating mechanisms, whether you understand it or not, this is what you're dealing with. That's what you actually need to resolve. Now we have a way to help resolve that. We have an additional tool to use. When is it horizontal transfer and when is it hybrid speciation? When is the gene transfer asymmetric? When do you have hybridization that does not produce a new species? How does that happen? What are the mechanisms that take place? And all of this has to do also with the issue of potential differences in hybrid fitness, which also are important for things like parapatric speciation, speciation by reinforcement, and potentially are important for making policy about conserving biodiversity. Because if we end up forcing species to live together that could potentially hybridize, we need to understand what the consequences might be. There are situations in which if you make two species hybridize, you're actually going to hurt both species. Okay. If the hybrids are always at a fitness disadvantage relative to the parents, every time there's a mating mistake, every time there's a reproductive mistake, that is a net loss of information from both species populations. And this could be non-trivial. Every time you make two incipient species come together and share information, you are reversing the evolutionary process. You are going to result in a net reduction in diversity. A net reduction in diversity is a net reduction in evolutionary options. So there are things that we may be doing in the name of conservation biology that are actually contrary to what we need to do to try to preserve evolutionary potential. We need to know what we're doing. We need to ask these questions. We need to ask questions about species concepts. And PACT analysis is an additional tool to help us understand what kinds of species we're dealing with. Because evolution produces a lot of different kinds of species. And they have different properties. They have different abilities to survive in different circumstances. For example, evolution produces a large number of peripherally isolated, highly specialized, highly localized species in cloud forests. And all of those species are evolutionarily ephemeral. All of those species, even without human beings on the planet, all of those species will last for a very short time, go extinct, and then new species will colonize the cloud forest when they come back again. So every time you have a change in the level at which the clouds come in, you have clouds covering the tops of the mountains and then they go away. Those clouds go away, all the species that are specially adapted to the cloud forest die. They all dehydrate and they die. Clouds come back, new species move into the cloud forest, differentiate, and they're also evolutionarily ephemeral. They're amazing, they're wonderful, they're beautiful, and they're not worth spending any money trying to save because we're not going to save them. Even if there's nothing in climate change that's due to human beings, climate change is happening, and isolated ephemeral habitats like cloud forests are simply going to disappear. And they'll come back again in a million years or something like that. But for now, any money we spend on trying to save the cloud forests is wasted money. It's money we can spend on trying to save something we could actually save. You have to know what you're dealing with. You have to know the history of the origin. You have to know the history of survivability of particular kinds of species in particular kinds of habitats. It would be nice to study actual character evolution and to ask whether or not there's certain characteristics that are transferred laterally more easily than others. There's a very interesting thing that Urs Satmar taught me about horizontal transfer in bacteria. Now, if you read some of the papers by people like Professor Doolittle, for example, you'll know that a lot of microbiologists are saying there is no phylogenetic tree of life. There's only a highly reticulated network because bacteria just exchange genes horizontally like crazy, and there's no pattern. Well, it turns out that that's not exactly true. Here's what really happens. If you take a population of bacteria and you starve them, at some point, they will digest their own metabolic genes as part of trying to survive starvation. If they get to that point and then you suddenly pour nutrients into them, they will come back alive, and if you then put any other kind of bacteria in with them, they will immediately obtain copies of the metabolic genes of the other bacteria by lateral transfer. And they will reconstitute their metabolism. Well, they will use someone else's metabolism, and they will go on their way. But guess what they never do? They never digest their own replicating material. The replicating material is always sequestered. They will die before they digest their own replicating genes. And they do not share replicating genes horizontally. So, if you're building phylogenies based on metabolic genes, like mitochondria, for example, and you're a microbiologist, the whole world looks like a reticulated network. But if you're using nuclear genes, if you're using not prokaryotes, but if you're using replicating genes, there's a phylogeny, is a phylogenetic tree. Which is true? Both. Which is more interesting? Seeing that whole picture, realizing that some genes can be transferred horizontally really easily, and others are really carefully sequestered. That tells us something very, very cool about one of the major transitions in evolution. And it also explains to us why we can get really different results if we not only just use different genes, but use different parts of the genome or different parts of the inheritance system. Okay? That means, right away, the first thing you have to do is to go and read everything that Nebraska has written about multiple inheritance systems and epigenetics. Because it's not just about the simple inheritance anymore. It's complex. And that all, for systematists, for evolutionary biologists, that all comes out of what can be transferred laterally and what can't. What gets shared, what gets protected. And you can ask that kind of question, what gets shared and what gets protected, at a lot of different levels. So let's jump from lateral gene transfer in bacterial metabolisms, and let's talk about primate social groups. Okay? Primate social groups. What gets guarded and what gets shared? Does anybody know? What part of the genome stays with the group and what part of the genome gets shared with other groups? All right, let me ask you another question. Or give you some data. In the province of Newfoundland, in the northeast of Canada, it's a fishing province, full of villages of fishermen. If you look at the telephone directory for each village, 90% of the people in each village have the same last name. But that predominant last name is different in each village. How does that happen? How does that happen? What is the last name? It's the name of what? Okay, my last name is Brooks. Whose name was that? My father's. Okay? In a fishing village, who stays with the fishing boats? Who gets the fishing boat when the father dies? The daughters or the sons? So who stays? And who goes to another village to find a husband? Same thing that happens in baboon groups and chimpanzee groups. The females get shared between populations, either voluntarily or not. Okay? That's interesting. Okay, there you have one pattern, one expected pattern of relationships among populations based on genes that have to do with being male. And that's going to look very different from genes that have to do with being female. So that means that if you're looking at mitochondrial genes, you're not seeing what happens to the males in the population. You're not seeing the whole story. You need to see this. Okay? So you need to know what's going on. And the whole issue of, that's why I said, it doesn't matter what level in the ecological or genealogical hierarchy you're talking about. It's the same kind of thing. A relatively simple phenomenon can generate a lot of complexity. So the reason you have these villages in Newfoundland where everybody has the same last name, but they're not all dying from inbreeding fitness depression, is because all the men in the village are closely related and none of the women are closely related. They're all from other villages. So if you grow up in a village where all the men are closely related, you don't want to marry one of them. It's like marrying your brother, literally. Or your uncle, or your cousin. It's like, you know, if the only dates you can get are people you see at a family reunion, then you're going to go look someplace else to have children. That is an intuitive recognition that there are two different patterns of genetic exchange and differentiation operating at the level of human biology. We understand so many of these things intuitively, and somehow they're not represented in our science. We've become so sophisticated that we don't know anything anymore. So, yeah, it would be nice to know something about this. Mitochondrial versus nuclear genomes. Mitochondrial genomes are only going to tell you what your mother and grandmother and great-grandmother and great-great-great-grandmother did. And as interesting as that might be, it might not be the whole story. Metabolic and replicator treatment. Now, are there some groups, and everybody has already knows the answer to this. Are there some groups where hybridization, hybrid speciation, is easier, is more common than in other groups? And if that's true, what allows that? You know, why is it that some groups show high degrees of hybrid speciation, other groups don't show anything? And it's not just the old prejudice that I grew up with. Oh, plants do this, but animals never do. That's crazy. Here's one of the places to start thinking about undiscovered high levels of hybrid speciation in animals. You go through the literature of cladistic analysis, especially cladistic analysis of invertebrate groups, like polychaetes, for example. And what you find is, oh, polychaetes are just a mess for phylogenetic analysis because I did a big study and I got 12,000 equally parsimonious trees and lots of homoplazy, and it was all over the place. Okay? If you're Vicky Funk and that's a group of plants, you say, I know exactly what's going on and I can tell you which ones are the hybrids. But you're a zoologist and you know that animals don't hybridize like that. So that can't possibly be the answer. Now, how do polychaetes reproduce? Once a year when there's a full moon, they just throw all the gametes into the water and go, woo! What are the odds that you're going to get some lateral gene transfer when you breed like that? Probably pretty high. Especially if you live in a highly productive area where there may be something like 30 species of polychaetes all living in the same tide pool. But this is a case in which we zoologists, have simply decided that this is not a possibility, so we don't even consider it. Okay? So, somebody should go back, look at polychaete phylogenies that have all this mess, reanalyze them using PACT, and find out if it's possible that one interpretation for the mess is that there's been a lot of hybrid speciation. Find out if you have a clade of 30 polychaetes and you remove 3 of them from the analysis and you only get 1 tree. Maybe that's going to tell us something about polychaetes that we never knew, that we thought was not true. So we need to ask whether or not reticulation is easy in some groups. We need to ask whether reticulation has happened more often in some groups that we think it never happens in. We need to ask about differences between prokaryotes and eukaryotes. And that goes back to the metabolic replicator gene thing. Because right now there's this strong belief that prokaryotes don't have a phylogeny. They're just a big soup of exchanging genes. Whereas, yeah, eukaryotes have a phylogeny. Okay? Well, both of those positions are probably partly wrong. We know with prokaryotes that replicator genes are not shared. And we know that with at least some eukaryotes, plants, the metaphytes, hybrid speciation is really common. And my suspicion is that there are groups of animals where hybrid speciation is much more common than we believe. All right? And then the classic botanist-zoologist argument. Okay? Maybe they're really not as different as we think. Maybe there's more hybrid speciation among animals than we believe. And if not, if it turns out that there really is not very much hybrid speciation in animals, but a lot in plants, what is it about those two different evolutionary systems? Because they diverged a long time ago, and they're both very species-rich groups with representatives all over the planet. So if in fact they are fundamentally different with respect to this kind of reticulated evolutionary relationship, why are they that different? Because it must be something that is really fundamentally different about plants and animals. Something at the origin of metazoans, something at the origin of metaphytes. It would be nice to know about that. That would probably be another one of the major transitions in evolution. Something that happened a long time ago that set an agenda more or less forever for a very successful group. So these are some questions that, this is a whole universe of investigation in evolutionary biology. And these are questions that we are not asking, in part because we have not been using methods of analysis that describe the real complexity of life accurately enough to make us aware that there's a problem that needs to be explained. We use methods of analysis that make us feel good. They make us feel safe. They make us feel comfortable. They make us feel like the world is so simple that we can understand all of it. Even though our daily experience is completely the contrary of that. I mean, everybody in this room has days or weeks when you feel like your entire life is out of control and you're not in control of any of it. That the world is so complicated and you're just one person, and how could you ever make a difference in anything? So, we're back. I know it's repetitive, and by now you're going, this is really boring, and keep saying the same thing. But it's really important, I think. It's really important for us to understand just how significant this evolutionary legacy of fear is. And it's because we're prey items. I mean, how many of you have actually seen monkeys, any primates, in the wild? You actually, I have. Anybody else here, have you ever seen monkeys in the forest? Real live monkeys? None of you? Oh my God. All right. Those of you who are not from Brazil who are watching this, they all raise their hands because all Brazilians know about their jungles. All right. So, you and I know. The rest of you, go find this out. There's a park here that's got primates in it. Wander down to that park at dusk, near sunset, and watch them. Primates are the most fearful animals in the world. It's partly because they're a lot smarter than rodents. Capybaras are a little fearful, but mostly they're just dumb. Okay? They don't know how frightening the world is. They really don't know. Primates are smart enough to realize that the world is full of things that want to eat them. So primates are like this. They're really like that. They're all like that. Now they're very curious, they'll want to watch. But the first thing they're going to do is make sure you can't get me. Okay, now I'll check you out. And we saw that, those of you who were in Puerto Rico, the first day we got to the field station, there was a little family group of bugios in a standard bamboo. And we went up, gave a little vocalization to let them know that there was a potential competitor there. And so sure enough, you know, Big Billy, the male, comes out to check out this guy with speaking bugio with a very bad accent. Speaking bugio with an American accent. Who are you? And he looked around, he checked us out, and you look up and the others are they're way up there and they're kind of looking around and being really quiet. He's checking. And he realized, oh God, it's just a bunch of biologists. They're not going to hurt me. But just to make sure, I'm going to let them know who's boss. So then he went into his male dominance display. He found a nice long branch where he was exposed. He stretched himself out so that I could see how big he really was. You know. And then just to make sure I knew it was his territory, went. That's really predictable behavior. But if I had done this, they would have gone like that. That's what primates are like. That's what we're like. That's why when we're given anything new, our first reaction is, no. Don't like that. And in science, we even say all new ideas in science go through 3 phases. The first is, you're wrong. The second phase is, you're right, but it's trivial. And the third is, you're right, it's important. But we've been doing that in my lab for 10 years. And that's no different than other kinds of primate behavior. So I really think that this is something that we inherit. And it's not maladaptive. I mean, the reality is, if some stranger walks up to you on the street and says, here, drink this, are you going to do that? Or here, would you hold this bag of white powder for me? You know? And then he walks away and a policeman walks up and arrests you for drug trafficking. I mean, you know, this suspicion, this fear of the unknown is a good thing in general. But we can't let it dominate our science, especially we can't let it dominate our science at a period of time when the most critical need for humanity in the next 20 years is going to be biology. You know, the physicists who make atomic bombs are not going to be the answer to any problems of the world in the future. If we're going to survive, if this is going to survive, it's going to be because evolutionary biologists break through into the public consciousness and say, here's what we need to do. And in order to do that, we have to be capable of saying, yes, the world is as complicated as you think it is, but we can handle that complexity. As opposed to saying, let me simplify the situation. E.O. Wilson once told me that one of the reasons that biodiversity does not get the kind of money from the American government as biotechnology does is because of the way biotechnology markets itself and the way biodiversity markets itself to the American Congress. You have to imagine, and there's an American Congressional Committee, a bunch of congressmen, they're all 60 to 80-year-old white men sitting at a table. And they're very powerful and they have a lot of money. And so a biotechnology guy comes in with his 3-piece suit and he sits down and he says, we are going to eliminate hunger. We're going to eliminate disease. We're going to make your children healthy and perfect. We're going to make you live to be 100 years old, and we're going to make you have a permanent erection, and you will be attracted to any 25-year-old woman in Washington, D.C. And all the congressmen are going, yes, yes, yes. But first, we need $600 million for research on Drosophila, and they can't write the check fast enough. Now, all of that's a lie. They cannot deliver on any of those promises. But it doesn't matter. They're saying what they want to hear, and they get the money. Biodiversity guys come in, and they got boots, and they're dressed like me, and they got beards, and, you know. They walk in and go, well, last year we came here and we said, you need to give us some money or things are going to be worse. You didn't give us any money. This year, things are worse. Give us some more money. And they walk away and they go, we don't want to hear bad news. We're not going to give any money this year either. So, and why is that? Because in general, the way we have tried to solicit money to support biodiversity projects is by making people afraid. But they're already afraid. They're Homo sapiens. They're fearful primates. You have to say, it's really complicated, but we have a plan. And it's not, I'm not saying that the solution to the biodiversity crisis is PACT. But I am saying that the solution to the biodiversity crisis is in the hands of evolutionary biologists. If we cooperate with each other, if we share information, if we're highly reticulated, and if we really want to confront this, if we are able to overcome our fears, then we can communicate that to the public. And we can communicate that to the people who actually have power. The people in governments and in international agencies that have the ability to say yes. And that's who we have to talk to. And we cannot make them afraid. Because they're already afraid even if they don't say anything. Look at pictures of Barack Obama from 4 years ago and now. I mean, he had no gray hair 4 years ago. Look at him now, he's a wreck. That job kills people. It chews people up and spits them out. And I think, personally, I think that's one of the reasons that in the last 2 months, his campaign has become very low energy. I think he's smart enough that he kind of went, do I really want to do this? You know, I've got a wife that I really love. I got a couple of kids who are really cool. You know, and every morning I hear all this stuff that nobody else in the world can hear. And it makes me afraid. And I can't say anything because it would create panic in the United States. I mean, you know, every day, you know, here's your toast, here's your coffee. Oh, by the way, here are the 12 people that were plotting to kill your family yesterday. You know, I mean, how would you like to live with that? Day after day after day. Okay, so these people, any policymaker who has access to privileged information is already afraid. They're already afraid. So we can't make that worse. We have to say, it's okay. It's complicated, but we have a plan. And nobody has been doing that. Conservation biologists have not been doing that because we've been acting like we're afraid. And so what you have is two groups of people who are afraid talking to each other. And all they do is make each other even more afraid. There's fight, 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 fight. And we have to break out of that cycle. So we have to start by saying things like, it doesn't matter whether this climate change is the result of automobiles and too many human beings. Who cares? And who really cares? What the ultimate cause was is not part of the solution. So why are we fighting about whose fault this is? Let's just solve the problem. In the United States, the one public person who has that attitude is Al Gore. And neither political party in the United States will support him. In all of the presidential debates, there was not one mention of climate change. Neither Obama nor Romney talked about it at all. Al Gore's name has not come up in the campaign for either party. The Republicans aren't attacking him, the Democrats aren't supporting him. Why is that? Because both sides know he's right. In the election that's happening today, in the state of Nebraska where I live, 70% of votes are going to go for Romney. And yet, I just got an email today from the U.S. National Center for Evolutionary Studies and Education in Evolution, saying the state of Nebraska is about to change its high school curriculum to include 2 critical modules on climate change. So here's a state that's going to vote for a climate change denier. At the same time, they're changing their educational program to make sure that their teenagers know how important climate change is. So they're not denying climate change because they truly believe there is no climate change. They're denying climate change because they have no answers and they're afraid. And if you're running to be the President of the United States, you cannot show fear. You have to be strong. So, everybody knows, all the politicians who say there is no climate change, understand that there is climate change. The branch of the American government that spends the most money every year planning for how to adapt to climate change is the United States Department of Defense. The American military is spending enormous amounts of money planning for how to adapt to climate change because they cannot afford to pretend there is no climate change. And a very politically, socially conservative state like Nebraska is adding climate change, explicit climate change instruction to the high school curriculum because they know that this is real. Everybody knows it's real. But nobody wants to admit it because right now, if you admit it, you look like you're afraid. And the people who are most responsible for putting the policymakers in a position where they cannot admit climate change without looking weak is us, is the biologists. We have put them in a position where if they admit this is true, they look like they're afraid because we've made them even more afraid. We have made this a discussion about fear, not a discussion about saving humanity, not about saving the future of humanity. And I come back to this on Friday, and I know I've already hit this before and I'm hitting it now, but we're going to come back to this again on Friday because this is really critical stuff. This is near time stuff. The next 20 years, the next 30 years, the next 40 years, things are going to change a lot. And that means that we have to start trying to change the discussion. We have to start acting like we're not afraid of what's coming. Hey, this has happened before. This kind of evolutionary change has happened before. We've got really good paleoecologists who understand what it's going to take to adapt to this. We can do this. That's how we have to act. And one of the ways to do it is say, we have a really powerful scientific weapon. It's called evolution. That's the most powerful biological mechanism we have for safeguarding biodiversity, including us. And that's why I'm so fixated on complexity. And how can we handle complexity rather than just ignoring it or denying that it exists? It's not really a joke when I say, don't be afraid of complexity. It's a bad joke now with that particular photograph, which was from one of the biannual conferences of complexity in Havana. which has now been shut down because the Cuban government was afraid of the discussions that were happening during these international conferences on complexity. Because one of the things that comes out of considering complex system dynamics is the more force you use to try to control a complex system, the more uncontrollable and unpredictable it becomes. So if you're the Cuban government and you're about to go through a transition, the best way you can ensure a peaceful transition is to ease all restrictions on your people. And they don't want to hear that message. And they don't want anybody else in their country hearing that message because they're afraid. And when people are afraid, they're violent. And we have to be responsible for saying, there's another way. You don't have to be afraid. We have a plan. And then it's up to your generation to develop the plan. Like I said, lots of research opportunities there. You know, I'm retired, have a good life. Okay, have a good lunch. Thank you very much. And let's hope America does the right thing today.